The organisation of DNA depends on the type of cell it's found in. In eukaryotic cells, DNA is found in the nucleus, although mitochondria and chloroplast also have their own DNA. Nuclear DNA is not actually visible until it forms into a chromosome, and chromosomes are only visible when a cell is about to divide, and you have to use a special DNA stain in order to see them. A chromosome is a combination of DNA, which has been wrapped around particular proteins, which I'll describe to you now. So in order to form a chromosome from DNA, you start with your double helix DNA, which then wraps itself around these little purple spheres called histones, which are a type of protein. Once you've got histones grouped together with DNA wrapped around them, it forms something called a nucleosome. And then those nucleosomes then coil around to form coils. And then those coils continue to wind themselves around into what's called supercoils. And then eventually those supercoils form your chromosome. The supercoils of nucleosomes are referred to as chromatin, and they form a cylindrical tube. Now I should point out that a single chromosome of DNA that's unreplicated actually looks like this once the chromatin has formed into the chromosome. What you're looking at here is actually duplicated DNA right before the cell is going to divide. So in fact you have two molecules of DNA here. It's number 1 and number 2. And they're joined together in a spot called a centromere. The nucleus of a human somatic cell, which is a body cell, has 46 chromosomes that form 23 pairs. Now 22 of those pairs are referred to as homologous chromosomes. And homologous just means the same, because these 22 pairs of chromosomes, the individual pairs are the same size, the position of the centromere in each pair of chromosomes is in the same position, and they have the same banding pattern. So you can see here that in this case we have 22 homologous chromosomes. You've also got 2 chromosomes which are non-homologous, and these are the sex chromosomes because they determine the sex of an individual. If an individual has one X chromosome and one Y chromosome, they will be a male, whereas an individual with 2 X chromosomes will be a female. The name for the remaining 22 pairs of chromosomes that don't determine sex are autosomes. Now when you lay out chromosomes like this in order from the longest here at number 1 to the shortest here at number 22, it's referred to as a karyotype, which displays the number and appearance of chromosomes in an organism. And the karyotype of an organism varies from species to species, and in some cases varies from individuals within a species. Ploidy is the number of sets of chromosomes in a cell or the cells of an organism. And when you're referring to ploidy, you use N to represent the number of sets of chromosomes. So in the example I gave you previously of the somatic cells in our body, you actually have 2 sets of chromosomes, one inherited from each parent. So this is represented as 2N, N being the number of sets of chromosomes, which in our case is 46 chromosomes in total. However, there are other cells in our body, which are the sex cells, so the sperm and egg, which only have one set of chromosomes. So N equals 23. There are different names for ploidy depending on the number of sets of chromosomes in the cell. If you've only got a single set of chromosomes, it's referred to as haploid, such as our sex cells, whereas our somatic cells are diploid. But there are in fact other organisms which can have 3 sets or 4 sets of chromosomes, or even more. Along the length of a chromosome, there are particular regions of DNA that determine particular characteristics or traits. They code for proteins, which are then expressed as traits in an organism. And these regions of DNA are referred to as genes. In a homologous pair of chromosomes, a maternal chromosome and a paternal chromosome, one inherited from your mother and one inherited from your father, these genes that code for the same characteristic, let's just say eye color, are found in the same location on both chromosomes. And these locations are referred to as locus or loci for plural. So in this image here, you've got 2 locus. Locus 1 contains genes A and B. So you can see that gene A, which codes for a particular characteristic, is in the same position on the maternal chromosome as it is on the paternal chromosome. And it's the same as gene B, whereas locus 2 only contains gene C. Alternative forms of the same gene in homologous chromosomes are referred to as alleles. So alleles are just different versions of the same gene. So for example, eye color. Eye color can be blue, brown, or green, and these represent the same characteristic but different alleles of the same characteristic. The reason alleles are different is because there's a difference in the nucleotide sequence between the 2 different versions of that gene. So an example in this image here is gene D, which actually has 2 different alleles represented by an uppercase D and a lowercase d. Similarly, gene E and gene F both have 2 different alleles. Now in homologous chromosomes, there's generally only 2 different alleles for each gene. In prokaryotic organisms like bacteria, DNA is organized into a single circular molecule, which can be seen in this image here. Now, because they only have a single DNA molecule, or one chromosome, they're actually haploid. Now, because prokaryotes don't have a nucleus, the chromosome is simply present in the cytoplasm, and the region in which the chromosome is found is referred to as the nucleoid region. There are actually smaller circular DNA molecules that float around in the cytoplasm, which are referred to as plasmids. You can see those here. Plasmids often carry non-essential genes and they replicate independently of the chromosome.